Good morning, friends. Today we will study types of antibiotics. What is antibiotic? The word antibiotic is derived from the Greek word anti meaning against and bios meaning life. So, as the name suggests, antibiotics means a substance which is against the life. When we see the definition of antibiotics, antibiotics are natural chemical substance produced by microorganisms which have the capacity to kill other microorganisms or inhibit their growth. So these are the substances which are produced by the microorganisms and which acts against microorganisms. Basically, we use antibiotics for killing the microorganisms. There are different class of antibiotics. One of the common examples are penicillins, cephalosporins, macrolides, aminoglycosides, and quinolones. Ampicillin and amoxicillin are the most commonly used antibiotic. They are a penicillin group of antibiotics. Cefixime, cefadroxil, ceftriaxone, cephalexin, these are the class of cephalosporins. Azithromycin, roxithromycin, erythromycin, these are the class of macrolides. Gentamicin, streptomycin, neomycin, these are the class of aminoglycosides antibiotic. And ciprofloxacin, ofloxacin, levofloxacin, gatifloxacin, sparfloxacin, all these floxacins are the group of quinolones. So depending upon the class of antibiotic, they have different mode of actions. Antibiotics can be of two type. Either depending upon their action, they are classified as bactericidal antibiotics or bacteriostatic. Bactericidal action means killing, which kills the bacteria, and bacteriostatic means which inhibits the stop or inhibits the growth of. Now we'll study how antibiotic works. Quinolones class of antibiotics. Which are the examples? Quinolone, ciprofloxacin, ofloxacin, gatifloxacin. These antibiotics inhibits bacterial DNA gyrase or topoisomerase enzyme which is responsible for DNA synthesis. So that is how they work. They prevent the synthesis of DNA by blocking the enzyme which is responsible for the formation of DNA. So there are two different kinds of enzyme. Either it is DNA gyrase or topoisomerase 4. So these are the enzymes by which they block and ultimately bacteria die. So this is the mode of action of quinolones. Another class of antibiotics like macrolides. Macrolides means azithromycin, roxithromycin, and tetracyclines and aminoglycosides. These are the antibiotics. They either binds with 30S or a 50S. Particularly macrolides binds with 50S ribosomes and aminoglycosides and tetracyclines binds with 30S ribosomes and thus inhibits the bacterial protein synthesis. So no longer bacteria prepares the protein and ultimately bacteria dies. Whereas penicillins and cephalosporins they binds with the bacterial cell wall and inhibits the bacterial cell wall synthesis. So there is no cell wall, no protection to the bacteria, and ultimately bacteria dies. So this is how bacteria antibiotic works. Okay, we need to understand some important terms. In antibiotic, two terms are very commonly used. One is MIC. What is MIC? MIC is a minimum inhibitory concentration. That means minimum amount of drug required to inhibit the growth of test organisms. MIC is normally written in microgram per ml and they are denoted as MIC 50 and MIC 90. There is a ratio of MIC 50 and MIC 90. What is MIC 50 means? Minimum amount of drug required to inhibit the growth of 50% of the test organisms. And MIC 90 is 90% of the test organism, and that is why they are always written in the ratio format. MIC 50 divided by MIC 90. That gives the minimum amount of drug shown the inhibit of growth of microorganisms. So that defines the potency of the antibiotic. Another term is MBC. MBC is minimum bactericidal concentration. What is MBC? Minimum bactericidal concentration to kill 100% of the test organism. Suppose there are 1000 organisms are there. So at what concentration antibiotic has killed all the 1000 microorganisms? So that gives the minimum bactericidal concentration. So that concentration is a concentration that has to be decided that at this concentration all these pathogens will be killed in the body. So that is the MBC. So that talks about the therapeutic window of the antibiotics. The another term is bioavailability. The proportion of active drug that is absorbed and therefore available to exert its pharmacological effect. In pharmaceuticals, bioavailability is a common term which is normally used. Say for any oral route of medication, suppose you take 500 milligram of paracetamol tablet. So when you take 500 milligram of paracetamol tablet, all this 500 milligram of paracetamol will not be available in the blood. Only 20 or 30 milligram of paracetamol will be available in the blood. So whatever the final concentration available in the blood is called bioavailability. So suppose if you are taking IV injection, in that case you will get 100% of bioavailability, because what you are doing, you are directly giving drug to the into the blood vein. So that is the final concentration of a drug which reaches into the blood vein. So there the bioavailability will be of 100%. This is very commonly used because all these different formulations have a different bioavailability. For example, tablet will have a lesser bioavailability, granules or powders or a syrup will have more bioavailability. So this is one common term. Another thing which you need to understand is area under curve. Look at the graph. On the x-axis we have given time and on the y-axis we have given concentration. So as the time goes, concentration of the drug increases into the body, but at certain period of time the concentration of a drug will reach to its maximum peak. After that, as the time progress, the concentration of the drug falls. So this creates one area, and this is called area under curve. This area under curve gives an idea about the amount of drug available to exert therapeutic effect. Now look at the another graph. Here we have given elimination or a T half. See, same axis, time as well as concentration. As the time goes, the concentration of the drug reaches to the peak concentration, which is denoted in red color. But after certain period of time, the concentration of the drug starts falling. So how much time it require to fall half of its original concentration, that time is called as T half. So this T half is also very much important, which also calculated how much time it will take to eliminate itself from the body. So my dear friends, with this we finish the basics of microbiology as well as types of antibiotics. Thank you very much.